Einen wunderschönen Montagabend zusammen. Have a nice Monday morning. I hope you had your breakfast, Stephen.
Yeah, but just finished.
That's a time difference. We have here in Germany, it's 8 p.m. as every Monday when we do our streams. Stephen is in Hawaii doing some research on Varroa, I think.
Yep.
And I met Stephen Martin in Hofgeismar on the Imkerzüchtertagung des DIP. And he held a presentation which I very much liked about basically the presentation you're giving today, perhaps some minor changes, some minor adaptations, where perhaps we just change to the slides and you explain what you're doing, who you are. I know a little bit, but it's difficult to explain in English for me. And just for those who don't speak English, diese Woche Stephen Martin mit einem Beitrag dazu, wie wir momentan Varroaresistenz vorstellen, wie das für Mechanismus funktioniert. Nächste Woche Steve Riley mit einem praktischen Ansatz, wie man diese Mechanismen in einer Zuchtpopulation im Prinzip in Basiszucht erreichen kann. Und in 14 Tagen werde ich eine Zusammenfassung beider Vorträge, den Vortrag von Steve Martin mit dem praktischen Ansatz in einer Kurzfassung bringen. Just for you, I just sketched up the next 3 weeks. This week, your talk about Varroaresistenz mechanism. Next week, Steve Riley with a practical approach to breeding of Varroa-resistant bees. And in 2 weeks, I will try to summarize your talks in German so that there is a complete picture about what we have this week and next week. And I'll try to go one step further. We have the program Varroaresistenz 33. You heard about in Hofgeismar, I think.
Yeah.
And the talker of the Varroa 33 project, Marie Förster, is also a member of this channel. And I will ask her to give a presentation what Varroaresistenz 33 is doing in the next years.
Excellent.
Okay. I talked enough. Now I will change to your slides and you will explain what you did the last years, and that's basically the culmination of a long research phase, what you're describing here.
Yeah. Okay, well, good evening to everybody on the YouTube channel, and thank you very much for inviting me. As we just said, I'm currently in Hawaii finishing up a 15-year study into Varroaresistenz, and I'll mention that a little bit later on. So what I'm going to talk about today is trying to give you an understanding about the Varroa-resistant mechanisms in honeybees, and it's going to be a global perspective. So just a little bit about myself. My name's Steve Martin. I'm now an emeritus professor at Salford University in Manchester in the UK. And that effectively means I'm a retired professor. I retired 2 or 3 years ago from teaching and research to concentrate on research, particularly on this Varroaresistenz. So I've been very fortunate to have been funded, and I've researched honeybees all over the world. I'm not going to go through all the different places, but you can see there's many places. I have quite a global viewpoint of what's happening to our honeybees. So I've been studying Varroa particularly for over 35 years now. I have done other things. My main interest are social insects. I've looked at termites and ants. I've also worked on chemical ecology of social insects and honeybees and varroa mites as well. But my main focus of research has always been the pests and pathogens of honeybees, particularly the varroa mite and its association with deformed wing virus. So first of all, this is a very old slide that we took, scanning electron microscope of a varroa mite. And I really like this image because what it does, it just gives you an idea of how highly adapted varroa mites are. And this has been the downfall of many beekeepers, thinking their bees are, very resistant and they'll deal with the mite and it's not going to be a problem. But they're highly specialized. They're very unusual for mites. Their body shape is different. And they only live in honeybees, the varroa mite. And so it's become highly specialized. Now, I'm not going to go through all the different adaptations that it's got, but one that I really like, it has a little thing called a peritreme, which is, let me just put this on it, highlight this here. It's this little organ here that pops out. And what they use this for is when they're submerged in the brood food, just before they start to reproduce, it's a little snorkel so they can breathe through it. So they can hide in the brood food from the bees, and then this snorkel allows them to breathe. So I really like this sort of high level of adaptation. And just one of the examples, I'm just going to give you a single example, is that one of the reasons that varroa mites can live in honeybee colonies is that they're chemically identical, indistinguishable from actual honeybee. And there's a couple of pests here. So Rolle, which unfortunately some of the older beekeepers will know about. It's a fly that used to live particularly on queens in honeybees. Once we started treating for varroa, it killed all the Rolle as well. It's a wingless fly, but if you look at the chemistry, this is a chemical barcode, these funny sort of peaks and troughs. And the top one is a honeybee and the bottom one is the parasite, either the Rolle or the varroa mite. And you will see that they are almost indistinguishable. So for us, we're visual animals, we can see there's clear differences. But in the dark of a honeybee hive, the honeybees use chemical smells. And so they can't distinguish between a Rolle and a honeybee, or a honeybee and a varroa mite. And this is one reason why varroa mites can wander around on the bees with complete immunity. And this is a picture of Apis cerana. So these are the native Asian mites, and native honeybees from Asia. And the mites, this is their natural host. And they're supposed to have really good grooming. But you can see here on the surface of the bees, there's lots of mites. This is a very heavily infested colony. And you would expect them to be groomed off. But in fact, they're not, because they're chemically invisible to the bees. And as I'll go on to explain, that grooming is not something that we believe that's very important in the resistant mechanisms. But this is just to give you an idea of just the level of adaptation that honeybees have and the mites have to get rid of, to avoid being detected. So one of the things that I did, and if you look here, this is an old paper from 1994 when I first started. I worked for the National Bee Unit, and my job was to basically come up with monitoring tools. And I spent many, many years along with other scientists, particularly a Swiss scientist called Donze, and we worked out their biology, the life cycle of varroa. And we know this extremely well now. We know when eggs are laid and the whole developmental period. And this has become very important in more recent years. And back in the 1990s, when I first started it, it was a very simple picture, is that lots of mites entered colonies, they sucked the bee dry, so they basically are feeding on their blood or the fat bodies. And the bees became crumpled, they had deformed wings. And this was pretty much what we believed. We did some experiments early on and found that this actually didn't seem to be the case. And cutting a very, very long story short, we basically discovered with Brenda Ball that deformed wing virus was actually a missing link. So this was able to explain just how a very small number of mites, about 2,500, can actually kill a large honeybee. So it wasn't the actual mites that were killing them directly by removing their blood or feeding on their fat bodies. It was actually this ability to transmit a virus, and it was a virus that was actually doing the damage. This took a long time to be recognized, but now I think everybody accepts that this virus is present in their colonies, and this is a big problem. Beekeepers can do very little about the virus, but it's not a problem, as we'll see, because you just have to reduce the number of vectors, get rid of the varroa mites, and the virus problem sort of disappears. And we now know retrospectively, but before Varroa arrived, deformed wing virus was an extremely rare virus. We only had a few cases of it worldwide that Brenda had picked up. Originally, it came from Egypt, so that was the first place it was discovered. It was originally called Egyptian bee virus, and we had cases in Belize and in the UK. But now Varroa spread pretty much globally. The virus is also global. It's all over the world. You can find it. There's still Australia. It hasn't been detected in Australia, but that's possibly just a matter of time. And now what's happening in your colonies in Germany and pretty much everywhere else in the world, we have two cycles of the virus, the deformed wing virus. We have low levels, which is a natural cycle. This is a natural virus, and this doesn't cause problems for your bees. It's been transmitted by the queens, by the workers in their food, can even attach to eggs. So it's not a good idea to move sperm around. And then also the big problem is where it's a mite associated. So when the mite transmits the virus by accident, and it's transmitted particularly into pupae, that shortens its length of life and causes it to die early. Some can have deformed wings, not all, a small proportion, and obviously they can't forage. And then it can get transmitted into the adults, but this is more of a chronic virus in adults. And so it's more, it doesn't cause such a big problem. It does affect things like time that they forage, but the adults become carriers, so it can persist in the colony for long periods of time. So the problem is that when Varroa arrived in every country, and the virus appeared usually 2 to 3 years afterwards, this caused most beekeepers pretty much to start applying treatments to control their Varroa populations. And particularly in the Northern Hemisphere, this has been the way to go. And this is what people have done. And so now we have, I think, almost 60 years of continual treatment in China, where Varroa first jumped the species barrier very early on in Asia. And they came in to control it very quickly, and they've been locked into that treatment cycle, particularly in America, where they're still suffering. I mean, last year, I think they had 55% of their colonies died, and they believe Varroa is the main problem. In fact, their 13-year average colony loss is at 40%, despite all this treatment. So treatments are now widely used across the Northern Hemisphere, but across the globe, it wasn't always the case. So there are countries, Latin America, Africa, the Caribbean, where Varroa treatments weren't used. This is either because they weren't available, or it was just simply they couldn't afford them. They were too costly. So it's not something that happened worldwide. And what's happened since is that now we have Varroa-resistant Apis mellifera honeybees. They've been around a long time in some countries, since the 1980s, particularly in Latin America. Originally was thought it was Africanized bees were the only bees that were resistant to Varroa. Later, it was discovered that African bees have become resistant to Varroa. And I was studying in the 1990s in Mexico. trying to understand what was actually the mechanism. It was very difficult, but it was very difficult to get funding because nobody wanted Africanized bees, and they thought it was solely linked to Africanized bees. However, more recently, there are populations that are well studied of Varroa-resistant honeybees in America and also in Europe. And I'll talk quite extensively about them. And it turns out that actually the mechanism is the same in all these populations of bees. So it's not specific to Africanized bees or African bees. It's in all bees. And as beekeepers, we often forget, we focus on the subspecies or the race that we're keeping. But in fact, as a scientist, I tend to look at Apis mellifera as a species, and all the races are just ecotypes that are locally adapted to that environment. So bees that are in South Africa or bees that are in Norway, as far as I'm concerned, they're just locally adapted honeybees of the same species. So they have the same underlying genetics and the same underlying abilities to resist Varroa. And a lot of people will say, well, yeah, this Varroa resistance, are the colonies very productive? Can they still do all this, like keep the mites down and be productive? So this is some work that I came across, and this is looking at the changes in the last 20 years. And so places where we've got resistance, or South America, there's been a real uptick in number of colonies, a doubling of their colonies, and their honey production is over at 300%. Again, in Africa, their honey production is almost doubled, and their number of colonies has increased. While we look at countries that are still struggling to manage Varroa, colony numbers are down in North America, and honey production is really quite low. And again, in Europe, number of colonies is down, and honey production is only up a little bit. If you look across into Asia, that's what sort of doesn't fit with this pattern, and this is mainly due to China. There's been a massive drive in honey production, and well, we won't talk about the quality of the honey, but they produce over 500,000 tons of honey a year, and vastly outstripping every other country, but the quality is quite low. And it'll be interesting to see what happens down in Australia with their production. But this shows that in countries where they are not treating for Varroa, because their bees are resistant, they're still very productive colonies, and they're doing extremely well. So I just want to give you two examples before I go on to explain. So I was very fortunate to go to Cuba a couple of years ago, and they have very good centralized data. So they have about 220,000 managed colonies in Cuba. They're European honeybees, so they're not Africanized, and this is because of the American embargo. So bees have not been brought into their countries for many, many years. They have Varroa. They have the Korean type, which is the same that's present worldwide, and they have deforming virus A and B. So they've got the two very common strains of the virus. So the situation in Cuba is exactly the same as in Germany or anywhere in the Northern Hemisphere. So these are not Africanized bees, these are European bees. The big difference here is 20 years, they've not treated for Varroa. And again, they have extremely good honey production, about 70 kilos. That's on average for all them 220,000 colonies. They get about 100 kilos in good locations. So we went there to study the mechanism to see what was actually going on. And then another population that's in Europe. So this is in North Wales. This is now, I've got to update this slide. It's now 16 years without any treatment. This is an area in North Wales. We've got about 500 colonies that have been monitored. They belong to about 100 beekeepers, and they've not been treating for over a decade now, all these beekeepers, but some of them, the original ones, they're up to 16 years. And again, we've studied these bees, see what their resistant mechanisms are. Okay, so for the bees, the beekeepers have always been ahead of the bee scientists. So for many, many years, beekeepers have been telling us that they have not been treating their colonies, and their colonies are doing very well. And it's only just in the last decade that the scientists have really sort of caught up. And to understand Varroa resistance, we really need to understand all the different components, and that includes the virus, because that's what really kills your colonies. Obviously, Varroa, because that's what transmits the virus. The honeybees, because people really concentrate on the different groups of honeybees. And it's important that if there's a mechanism, that it really needs to be widespread to the species and not to the strain, because if, say, I found that buckfast bees were resistant, only buckfast bees were resistant, then that wouldn't solve the problem for other countries. Beekeepers, it has to be, whatever resistant mechanism, it has to be adaptable for beekeepers to use. And then we'll talk about bee behavior, because that's at the heart of all this. So first of all, we can now pose a question: what happens if deforming virus is absent? And there are a very small, unique number of cases where we've actually discovered this. So there's a very small island off Brazil where they've had varroa mites, they have European honeybees, and they've had constant mite levels for about 25 years. The levels are very high, so there's about 15% of the adults are infested and 20% of the worker brood. They're Apis ligustica, and there's no colony losses on this island, or nothing due to Varroa. In fact, the beekeeper, Lydia, there's the one with the pink trousers on, she's in her beekeeping equipment. She doesn't wear a veil or anything. The bees are very quiet. She doesn't even know about Varroa. And what we discovered on this population is that actually deforming virus is absent, and the bees have become resistant. Now, when we did this study, we didn't know about the new resistant mechanisms, so we weren't able to sort of study that. But we do know that their ability to reproduce new offspring was extremely low. And that fits with what I'm about to tell you. That was a single study. We now have two other studies which have sort of reinforced this idea. So deforming virus is absent on the Solomon Islands. Again, we have Varroa resistance there. And on Papua New Guinea, in the highlands, we have Varroa resistance there. And again, deforming virus is absent. These are really small populations, very isolated. And this is something that is not something that we could ever achieve. We could never eradicate deforming virus from our honeybee population. It would be as difficult as getting rid of COVID from the human population, and we've spent trillions of dollars on that, and it hasn't worked. So this is just a scientific interest. It is quite interesting for Australia, because at the moment there's a possibility there's no deforming virus on that island. So the longer that lasts, the more chance that the bees have of becoming resistant if they don't treat. But I think the beekeepers are already starting to treat, which is not the right thing to do, but I'll explain why later on. So that's all I'm going to talk about on the virus. What's really important, or the big difference that we've known for decades now, is in resistant honeybees, the reproduction of Varroa is quite low, while in susceptible colonies that need to be treated because otherwise the mites would kill the colony, reproduction is better. And this is been the sort of paradox. We've not really understood the mechanism here of why in resistant colonies mites don't reproduce very well, where they reproduce much better in susceptible colonies. Fortunately, now we seem to have worked this problem out. And I'll cut straight to the chase. So this is basically our currently working framework of what actually goes on. So it's a type of hygienic behavior. And I've got to be careful, because when I talk about hygienic behavior, people will think about freeze-kill brood or pin-kill broods, because hygienic behavior is sort of being hijacked by these two methods, but it is only one part of hygienic behavior. Hygienic behavior is a general bee behavior which can detect diseases, things that are wrong in their brood, from eggs through to pupae, and then they'll remove them. So it's a generalized behavior. And so this hygienic behavior I'm explaining here is specific for Varroa. And so what appears to have happened is that the bees have learnt to smell the odors of the mites, or it actually seems to be the mite offspring, and specialized bees can do this. And when they detect these odors, they then uncap the cell. So they make a hole in the cell. We know the hole is slightly bigger if it's mite infested, and it's smaller if there's no mite in that cell. We think that the holes may be made just so they can detect better what the smells are. So now we have bees that come along, they detect the smell of the infested cells, they open up, they make a hole, and now these cells with these holes in, there's two options. One, these cells can then be recapped. So basically a bee will come along, see that there's a hole in the cell, check that the pupa is still alive. If it is, they just then seal the cell over, even if it's got mites in it. Or the other option is when these cells are opened, that the bee will come along, detect that it's an infested cell, and then basically cannibalize that individual pupae down. Unfortunately, the mother mite, being highly specialized, and as I mentioned before, chemically mimicking the pupae, can't be seen. She's very agile, so she escapes from the cell so she can reproduce again, but she loses all her offspring. So all the eggs and female and male offspring she's laid in that cell, they all die. They get unable to survive outside the cell. So she loses them offspring. And that's a big blow to the mite because they can only reproduce 2 or 3 times in their lifetime. So they're losing quite a lot of their offspring during this procedure. And this, when the cells are recapped, it doesn't happen once. Then these bees can come along again, these specialized bees, and uncap them again and go through the whole cycle. And I think Ralph Butler, the most he's ever observed is 17 times a cell was opened and closed during the developmental period of the brood. And every time the cell is opened, there is a chance that it's going to be detected by a bee that will then cannibalize it. So this is the sort of framework we use. So I'll now go on to the evidence that we have for this. So first of all, beekeepers in Europe and elsewhere around the world, ever since Varroa appeared, have seen an increase in what we call bold brood. And this is where you have these pink to purple-eyed pupae. They're uncapped, often in clusters, and beekeepers call them bold brood. And often they say this is a bad thing, a failing queen, there's a problem with the queen, and requeen it. We now know this is actually a good thing. This is the evidence of the bees looking for infested cells. They also do this for wax moth. And we think that adaptation for looking for wax moth, where they uncap and recap cells for wax moth, has been adapted for Varroa. And the way that we look for this is for these recap cells is a little bit more tedious. And basically what we'll do is we'll carefully remove the cap of a pupae. We'll flip it over and look at the underside. And on the top right, you can see the underside of the cap. It's all very shiny. And this is the silk cocoon of a pupae that's not been disturbed. But the 2 lower pictures, you can see where a central area has been cut out and then refilled with dark wax because the pupae skin, the cocoon, has been removed and it's filled with this dark waxy material. From the top, it's not often, it can be impossible or it's difficult to observe it, but from underside, it's very clear. And then we call these recapped cells. And it's evidence that at some point, the cell's been opened by the bee and then recapped over. So we can use this to see if a population can detect infested cells. So when we look at the amount of recapped cells in Varroa naive populations, so these are populations that haven't got Varroa, like this is Australia, a couple of islands off the UK, we can see that recapping is a very rare trait. So we rarely see it in colonies without Varroa. When we look at susceptible populations, and this is work from Melissa Odey that studied populations in Norway, Sweden, and 2 populations in France. that were all treated, and we looked at populations in the UK. The yellow bars are non-infested cells that have been recapped, and the red bars are infested cells that have recapped. And the first thing to notice is every population, the red bar is bigger than the yellow bar, which means that they are targeting infested cells, because there's much fewer infested cells in the colony than non-infested cells. So they're able to detect these cells, but at the maximum, somewhere between 10 and 20% of these cells have been investigated. Now, if we go to look at a population of resistant populations, and again, we're comparing Melissa Odie's populations in Sweden, France, Norway, two in France, our studies in England, Cuba, the work I've been doing in Hawaii, and then Africanized bees from Brazil, and then African two races there, the Scutellata and the Capiensis. You can see that the resistant populations are much, much better at targeting these infested cells. In fact, some of the ones here in Hawaii, they're about 80% accurate at getting infested cells. So 80% of infested cells at some point get opened up and exposed to bees. You'll also notice that there's a big increase as well in non-infested cells being opened. So it looks like they're making lots of mistakes at the same time. And we can now explain that is that recapping, this behavior, is actually spatial. So they're clustered. So the recapped areas tend to be clustered around infested cells. And this is some work done by my PhD student, Isabel Grindrod. And we think what's happening here, it's very difficult for the bees to detect, even these specialized bees, which cells are infested, where the smells are coming from. And we know from a French group what these compounds are. And so they know the general area. And so they start opening cells to actually try and find which cell has the mites in. And so you end up with these clusters, as she showed quite nicely. And so basically that's the recapping side. So now we're going to look at the evidence for this removal side. So when we've looked at removal, and this is what a beekeeper hopefully will see in a resistant colony, and we do, it's not very common. You have to just catch it at the right time. But the picture on the left-hand side of your screen there is of a infested pupae. I know it's infested because you can see, I'll just point here, just there. On the side of the wall, there are two white dots, and this is the mite feces on the wall of the cell. And so we know this is an infested cell. And then there we have the pupae that's half cannibalized down. And in fact, there on the wall there is actually a mite offspring. So this is an infested cell which has been eaten down. Now on the the right-hand side of the screen, so this is some data from South Africa, Hawaii, and Cuba of these populations are removing between 50 to 80% of all infested cells. So we artificially infest these cells, and then they're detected and removed by the bees. Treated populations, it's much lower. It's about 20%. So this is the big difference, where these resistant populations are just better at detecting and removing them than actually the treated populations. And what we think is happening in the bees is it's quite this hygienic behavior, and this is from Marla Spivak's work, is a complex behavior. So we know from the French that the bees, these highly specialized bees, we'll call them sniffer bees, they're detecting a group of ketones and acetates, which we think are coming from the mite offspring. Could be coming from the infested pupae, but I think it'll be the mite offspring. Between 15 and 20 days old, a very small number of these bees have very high levels of a neurotransmitter in their brain. And so they're very sensitive to smells. And so these are the ones that go around and detect the smells. Then we have these remover bees. These are the bees that will cannibalize down. They are less sensitive to the smells, but can still detect them. And as long as the cells open, these bees will then cannibalize them. And then we have the recapper bees, which basically are very insensitive to these smells. So they really don't detect them. And they can't discriminate between a cell that's got mites and that doesn't have mites. So they just recap the cells over. And so that's why we think that you've got these at least 3 groups of bees that do this, involved in this behavior. And so all the mite resistant traits that have been known about for a long time, we think they're all linked together. So effectively, if you start, you get increased mite detection, and that leads to 2 traits that we can see and measure. One is increased recapping, which we think is a proxy. So it's just doesn't directly cause a resistance. It's just something that's a good indicator of it. Might have a small effect, but it's only small. The main effect is increased mite detection increases brood removal, and that's what people call VHS, Varroa sensitive hygiene. When you get an increase in brood removal, this decreases the abilities of the mites to reproduce because they're losing these offspring, and that's called suppressed mite reproduction. So one leads to the next. If you reduce the number of ability of mites to reproduce, you decrease the number of mites in the colony, and that decreases the amount of deformed wing virus in the colony, and eventually that leads to increased colony survival. I just want to quickly go through this long-term study we've been doing here in Varroa, because it's gone through the full cycle of what people see. We're very fortunate that it's a natural laboratory. So 2 of the islands, Oahu and Big Island, have Varroa present, and the other islands are still, Varroa is absent. So we have this nice control setup. And trying to summarize everything into one slide is this. So the story, very briefly, is in 2006. We had a large managed population, and this is on the island of Oahu, where we've done most of the studies, and we have a very large feral population. In 2007, Varroa arrived in the country on the island, and within 6 months to a year, the beekeepers had lost about 65% of their managed colonies. The feral population started to decrease, and basically between 2009 and 2013, all beekeepers were treating their colonies with some sort of, mainly, or 90% were using chemical miticides. And then where there was no selective pressure, obviously, because the beekeepers are controlling the mites. However, in the feral population, there was extremely strong selection, and their bees, the number of feral colonies collapsed. Almost they all disappeared, and there was very few that remained. However, if we move on to about 2014, 2019, we started to see the feral population started to recover, things started to come back, and a small number of beekeepers started to collect bees from the forest and stop treating them, and realized that these wild swarms actually appeared to develop resistance. Most of the beekeepers were still treating, although they were also collecting swarms, but didn't realize that they were resistant. From 2019 onwards, we did some experiments. We were able to show that the bees now on Oahu had become resistant. They had this high removal trait, the high detection, and the high recapping, and they also very low ability to reproduce, so they had a high infertility, basically. So they were running out of eggs. We informed the beekeepers, and now in 2023-24, about 82% of beekeepers on the island here don't treat. So they've got widespread non-treatment. Their colonies losses are just, actually they're better than the mainland, and the feral population has recovered quite nicely. So what we think happened in the feral population is initially, if we start at the increase of mite detection, that'll be quite low. So we'll have low mite removal. There'll be variation in the population. There'll be some increase in mite infertility. There'll be obviously some reduction in mite growth. And then we come to the crucial stages. Is there enough mites in that colony or enough virus for that colony either to, well, it calls it to die or call it to survive. And initially, most of the feral colonies would have died, and a very, very few of them would have survived and been able to go around this cycle again and to get better and better at this. And what's happened now, if we sort of jump ahead 10 years, this is what we can detect now in these populations, where we have the increased ability for mite detection. So they're detecting 80% of the cells from the recapping. They're removing about 65% of all these infested cells. The infertility of the mites is really quite high now. I think we're looking at about 80%. It's a little bit high actually, but we're looking at a high infertility. And then that means that the number of offspring are produced is about 0.6 to 0.7 mites per cycle. So we've got real reduced mite growth, and that reduces the number of virus. So very few of these colonies now die. Most of them survive, and this cycle continues onwards. And so over a long periods of time, we've got some nice data from Mexico and Brazil, where we've been studying these populations for nearly 20 years. 20 years ago, the average infestation level of worker brood of resistant populations was 20%. They get better and better at this removal and detection, and now they've driven the mite populations down to about 4% of infestation in brood. We don't think they'll go to zero. There's no real need for them to go that low. Most of these pests never get eradicated, but they'll get them down to where they really cause very, very little damage, which is what's happening now in Brazil. And I think the infestation level here is about 6%. I'm sure it'll fall a little bit lower over time, but the beekeepers are not having to treat now, and the bees are doing the job for them. And I just want to quickly finish before I give you some advice. On the UK, we've got some really good data that's coming from the UK of the effects of non-treating. So currently in the UK, about 25% over the last few years of beekeepers are not treating. So that's around about 7,000 beekeepers. So it's quite a good proportion. We would like to see that increasing. And there's about nearly 2,000 of these have not been treating for a long period of time, about 6 to 15 years. However, it would appear that the people that are not treating suffer slightly higher losses, between 5 and 10% over the last 3 years. And most people will suggest, oh, well, that's because you're not treating for Varroa. But when we looked into the actual data, where the beekeepers give for their reasons for losses, Varroa doesn't actually figure very highly at all. In fact, it's actually slightly lower in treatment-free beekeepers than it is in people that treat, but it's well below 10% for the 2 years we have data for. The bigger problems tend to be things like starvation, queen issues, and weather, which I'm sure they're sort of all related. And there was some nice data that I saw coming out from Sweden, where basically they were showing that the colony losses are directly linked to how much feed that were being provided over the winter. So I think it may be something that's linked to this, where beekeepers are maybe not monitoring their colonies quite as closely as people that are treating, and therefore they're not picking up on things like they're running out of food. So it's something that people who are not treating need to be a little bit careful of. And then the other thing that's quite surprising, there's some ideas that if you want to have resistant bees, then you have to do this in isolation. You have to breed these on isolated islands. But a survey which we've done over 3 years now, actually over 4, looking at the proportion of people who are treating and non-treating at whatever scale all over the England and Wales, we have postcodes or areas where we have both treated and non-treated beekeepers in the same area. We're not sure why this is the case, but this is the data. And we're not sure whether the feral colonies are having a bigger effect than we're expecting. The effect of drones maybe not that important. We really don't know on this front. but it does seem to be that you can have both treated and non-treated beekeepers in the same area, and both seem to be doing their own thing. We did, to help the beekeepers, very briefly, we did a queen swap experiment where we just swapped queens from that were from resistant colonies into non-resistant colonies, and vice versa. And I won't go into this in any real detail, but the results were that the queens, if a locally mated resistant queen, if it's put into a non-resistant colony, so colony that has workers that are non-resistant, they will become resistant. So the genetics is in the queen. So whatever it is, the mechanisms, this hygienic behavior that the workers conduct is actually hardwired into the queen after several generations. The mechanism of this is almost certainly an epigenetic mechanism. So this is why it happens very quickly over a few generations. This is not unusual. These type of mechanisms are very important when it comes to environmental responses. It'll take a few years to work out the actual details, but it's really good for queen rearers and people who are trying to select. So you should be able to, if you get these queens, locally mated queens, assuming they've been mated with a locally mated drone, replace them into any colony, and you should have that colony should become resistant. So with the queen swap experiment, we worked out with resistant drones mated with the resistant queens, you've got resistant colonies when you use the queens. Susceptible drones, susceptible colonies, you had susceptible colonies. We don't know what the role of the drones are, and we applied for some funding, we didn't quite get that. So at the moment, the role of drones is sort of unknown. People can estimate or guess. Because it's an epigenetic mechanism, drones could play a really important part, or they could play a very small part, but this is something that we really could do with working out. So this is one of the gaps that needs to be filled. So the big problem with Varroa, particularly in the Northern Hemisphere, is at the moment that people that are keeping resistant colonies are going through this Varroa treatment cycle, where effectively you have susceptible bees, you need to have treatment. So you're not, it means the bees are basically got a very low resistance to Varroa because there's no selection for Varroa resistance. So you just keep going round and round in this cycle. While the Varroa resistant cycle, where you've got bees that are resistant to Varroa, you don't require treatment, and they get better and better at getting resistant. And so for beekeepers, you need to be able to, who want to give up the idea of putting treatment. And I think nearly all beekeepers are not comfortable about putting chemicals into their colonies, need to change from this Varroa resistant cycle, Varroa treatment cycle into a Varroa resistant cycle. And the two big things is the fear of change, and then the fear of loss of money for commercial guys, and then loss of colonies for people that are, or maybe only have a small number. So there's several options that I'm going to highlight, but the main message is do not stop treating your colonies, because if you're treating and you suddenly stop, the chances are that these colonies will die because they won't have sufficient resistance there. So it's a transition that you require. And so the first one is you can't try and collect swarms from long-lived unmanaged colonies. This is quite a common thing that's been done by many people that have moved into keeping resistant colonies. So there's a good chance they'll be resistant, but you can't guarantee it. So you'll need to keep an eye on these colonies. Oh, I've got a little bit of... The other one is if you've got somebody who's already got resistant bees, you could requeen from a friend with resistant bees, and that's been done here on the island and elsewhere. But you need queen rearers that have these resistant queens. Oh, sorry about my slides here. So collect swarms, and then the final one. I should have checked this. Right, here we go. And the last one is keep looking, monitor, and select and transition off your treatments. So this is basically where you start to select from your own stock. It's better if you get together as a community, maybe do it as a group apiary. There's a lot of people I know in Germany trying to select. There's the 2033 project where they're trying to speed up this process of selection, and then hopefully they'll be able to sell the queens on. There is one group that in the UK, Steve Riley, who's talking next week to you. He's got this lovely book called the Honeybee Solution to Varroa. They've gone ahead, they've selected for their colonies, and now they don't treat. And he's got a very nice step-by-step guide. I won't go into this in much detail, but it took them just over 5 years. And they just used mite counts and other factors, such as looking for evidence of these pupae being removed on the bottom boards. And very simple things. So they just used things like they've got 4 colonies here. They're looking at the number of mites were dropping in the colonies, and they bred from the 3 colonies with very low mite drops, and the one that had high mite drops they didn't breed from. Euthanized the queen, and they just did this repeated year after year. But Steve will explain this in much better detail. And so there's lots of resources out there. There is I've done a special issue which is just for beekeepers. It's just to outline pretty much what I've talked about today. There's Steve Riley's book. There's a really excellent book. Unfortunately, it's in Polish, but we are at the moment translating into English, so that will be available hopefully by the end of the year. The beekeepers asked us to put a lot of this information on a website. So there's the Varroaresistenz.uk website, where nearly all the scientific studies that based our research on from myself and others is free to download on that. There are case studies and advice for there. And also Melissa Odie, myself and Marla Spivak are hoping to write a book on called Resisting Varroa. There's a website, Melissa's doing a Kickstarter. So just a shout out for Melissa. So if you visit that, register your interest, and we'll see if we can make that book a reality. So there's lots of resources out there for you. And then finally, all this work that I've been doing over these many, many years, particularly these last few years, trying to sort of help beekeepers globally, and this includes China, though that will be a real challenge, to transition away from treatment, is basically nearly all funded by beekeepers, particularly from the UK, and it's generated lots of other bee scientists. And so I've got to thank them for the funding, allowing me to travel. Currently, I live in Hawaii to finish off the work here. We'll get that published, and we'll write some articles for the bee press. So thank you very much for listening, and hopefully I'll be able to answer any questions you have now.
Thank you very much at this point. It was a very dense presentation, and 40 minutes for a very interesting talk you did. And indeed, we had some questions. First question was a little bit back when you started, and you already addressed much of it. It basically was a question: is the breeding approach non-treatment and raise from the surviving bee stock?
That is one approach. It's quite a brutal approach. If you have enough stock to do this, you are going to probably encounter large losses at the start. And one thing that's really important, if you are going to go down this line, is that you need to monitor your stocks and not allow the non-resistant colonies to collapse. Because what happens then is if the mites will just migrate into the more resistant colonies and will then overwhelm them and kill them. So once the mite populations are going up in the colonies which are non-resistant or less resistant, you need to euthanize them and kill them colonies to stop mites drifting into other colonies. This can be a substantial problem within an apiary, or you move you have 2 apiaries, or you treat the colonies that the mite populations are increasing in rapidly. So it is a possibility you can do this, but you must not let the colonies collapse. I've measured colonies which are resistant and have been overwhelmed. We know if you get more than 40% infestation rate in a colony that is highly resistant, it will kill itself by removing well, it's going to remove 40% of the brood because of all them infested cells, and then the colony will just collapse under its own weight because it doesn't have enough bees to survive. So it is a possibility, but it's a more brutal approach to select from that. People have done it, but you've got to get rid of them excess mites. So you need to increase your monitoring.
I actually had this experience once. I introduced a highly resistant queen in a highly infested hive, and it collapsed. I wasn't prepared for this. It is what was a little bit late in the year, and it took some thinking to get to the reason for the death of this colony. But this happens. The next question was, is the brood damaged by the recapping process?
No. The brood will develop completely normally. In fact, I think Steve will talk about it, but if you go into the colony one day and see the ball brood and then photograph it and go back the next day, you'll find it's recapped and you wouldn't even know which ones had been recapped over. So the brood can develop completely normally, but it doesn't affect them at all.
Okay, one question. But this, I don't exactly understand the question. The mite is very close to the center wall at the beginning. Can the mite also be reduced with specially treated mite middle walls?
I think this might be so. The male to the first egg that's laid is the male egg, and that is very close to the top of the cell.
Okay.
Maybe this, and when I first saw Melissa Odie's study on recapping, I suddenly thought, oh, I know exactly what's happening here. They're going to open the cell and remove that male egg, and if they do that, then there's no mating, and so everything becomes infertile. However, I've spent a lot of time trying to look for this. Well, I have looked for it, and this is not what the bees do. This is another good example of what we think the bees should do and what the bees do are two completely different things. So the recapping process doesn't seem to affect, there's no increase in male death that we could detect in all our studies. So the recapping is only opening up the cells to allow bees to cannibalize the entire pupae down. If they did just kill the male egg, it would be way more efficient because they wouldn't have to then actually destroy the pupae. But that's not what the bees seem to do. So I think that may answer part of that. Treating, you could maybe treat your colonies by spraying things inside the cells, but I don't think that would be a good idea, to be honest.
I don't think we need more treatment plans.
No, we don't.
Actually, I didn't quantize this, but when doing SMR assays, like Harbor assay, we actually monitored, or at least have seen cells without a male, but otherwise normal brood development of the mite. But I don't know if this has a big effect in the whole process. If that's
it does. So male death is part of the infertility measure that we use, and it's not uncommon. These mites are very, very unusual in that they only produce one male per reproductive cycle. So if that male dies, then none of the females are then mated, and so they become infertile. So the reason for the males dying, we don't know. Sometimes they fail to get down to the feeding area, which is lower down at the bottom of the cell, and so it dies at the top of the cell. It's not a huge part male death. I mean, 10-20% of the males can die. It doesn't seem to increase with the recapping in the resistant populations. It's something that we've always, it does contribute to them. So yes, you will find what look like missing males. I'm almost guaranteed that if you look hard enough, you will find that dead male, but it could be a dead egg or a dead protonymph, and they're extremely hard to find and without a lot of time and expertise, but they're normally there. But yeah, they do. The males do die naturally for whatever reason, we don't know, but it's usually between 10 and 20%.
Okay. Next question is about the pin-killed brood. Here's somebody who is trying to select bees by pin-killing brood and asks if this might be futile to do.
To me, yes. This has always been a bit of an anathema, as in that freeze brood kill and pin brood kill tests, you are selecting bees that are very good at detecting and removing dead brood, because that's what you're doing, you're killing the brood. Varroa does not kill the brood. This is why it actually survives. If it does kill the brood, and this does happen with one of the viruses, so acute paralysis virus, if it transmits this virus, it kills the pupae, but then the mites can't reproduce. So the mites die as well in the cell. So it's vitally important for the mites not to kill the brood, and also the virus. And this is why deforming virus is one of the one that's come to the fore. So there's been a long-term assumption that highly hygienic bees that are good at detecting pin kill test or dead brood will also be able to detect Varroa. There is not as good scientific evidence that these things are correlated. So when we look at resistant populations, they're not particularly good at removing dead brood because they're not been selected for this. So they're looking for different sets of chemicals. So this is just something that beekeepers can do and beekeepers have done for a long time. But for me, there's no logical sense. The two are not connected because chemically the bees are looking for different scents. And there's very little hard, good scientific evidence that if you've got bees that are very good at pin kill test, they're very good at removing Varroa. It seems to be two different traits.
Okay.
Because they're going for two different chemicals.
Okay. A question about exactly those chemicals. Is it really the smell of the mites or the smell of the virus, probably deformed wing virus? And the question continues because dogs can smell types of cancer. So there are possibilities for animals to detect smells we can't even think about.
Yep, that's a really good point. So we think it's the smell of the mites. The work that's been done has been done on infested. It comes from infested brood, so we cannot rule out 100% that it doesn't come from the actual pupae. But we've done some preliminary chemistry which suggests it's actually coming from the mites. And the compounds are fairly unique to the compounds are unique to bees. So you don't usually find these compounds in bees. Now, most of the individuals in a colony are infected with virus. So the levels of virus are higher in infested pupae. So that is a possibility, but we'd expect it to be much more widespread. The amount of virus, it's much more likely to be the it's more likely to be the mites that are producing the compounds, and it's simply an experiment. So when we take out the infested mites, we can chemically wash them and have a look, and this is where we find large amounts of these ketones and acetates. Well, then we will look on the pupae of a non-infested pupae, although it still has virus at fairly high levels. We don't detect these compounds. So it does look like it's the mites and not the virus that's producing these compounds.
Otherwise, it would be a bad message, because we would need high virus loads to have the mites removed.
Yeah, and that would further complicate things.
But it's good to know that current scientific reasoning is it's the mite.
Yeah.
One last question, and then I would close the session for this evening. This question already has been asked twice. I'll try to find it, otherwise I will just refer it to you. Basically, it's do you already have insight into the resistant mechanisms of honeybees against the Tropilelaps mite? In other form, do you expect Varroa-resistant mites to be better prepared for Tropilelaps than non-resistant bees?
That's a really interesting question. So I was recently working in China, and there Apis cerana has no problem with Tropilelaps, even though Apis mellifera does. Now in China, there's no resistance. I suspect that the same thing will happen. I have no idea whether the resistance to Varroa will give it an instant resistance to Tropilelaps. I suspect not, because it's going to be yet another chemical message, a chemical difference that they have to learn. So resistant colonies may already have an advantage, and it may take them less time to learn this. I suspect that they will become resistant to Apis, to Tropilelaps. It may take some time, but if you've already got resistance, I think you're already hedging your bets, because you're going to have more of these specialized sniffer bees, which are the ones that are doing that initial detection and removal. But I think they are going to have to learn new odors that are coming from the Tropilelaps mites. But we've got a real good head start from the science, because we know one mechanism towards a mite, and we can use the same methods that we've used, like the French group for the chemistry, and to see if it's different or the same chemicals. Highly unlikely to be the same. So I think there's just going to be, there'll be a delay. A good example is small hive beetle. So here in Hawaii, small hive beetle was a real problem for a couple of years. Now small hive beetle is in pretty much every colony, but it's not a problem at all. And people don't use beetle blasters. There's no treatment. The bees have learned to adapt to the small hive beetles very, very quickly. And I think they'll do the same thing for Tropilelaps, like they've done for Varroa, as long as, and it'll be quicker than the resistant. But if we start treating, we'll be locked into that same treatment cycle. And Tropilelaps is worse than Varroa. It reproduces much quicker and can cause a lot of problems. So yeah, let's hope the bees learn quickly for Tropilelaps. I'm sure
bees
they
learn quickly. Thank you very much. Just as an example, the feedback in the audience is good. They liked your presentation very much, the information you gave us. This leaves me with a thank you very much to you, and have a nice day in Hawaii. Your day is way longer. You just started your day. We are late in the evening.
You go to bed.
And I know I was very confused when we tried to establish when we have to meet in time in this.
Yeah.
And the last thing I will do is to repeat for the audience what we will do on the channel the next weeks. And I will do this again in German for those who skimmed to the end. Heute hatten wir Stephen Martin. Nochmal herzlichen Dank an ihn mit einer Skizze über den Varroaresistenz-Mechanismus bei der europäischen Honigbiene. Nächste Wort Steve Riley. Ihr habt sein Buch in dem Vortrag von Stephen Martin bereits gesehen. Er wird wesentlich weiter ins Detail gehen, nach welchen Zuchtmechanismen er arbeitet, was er noch zusätzlich an Hintergrundwissen über dieses Verhalten in der Varroa in den Zellen hat. In der Woche darauf mache ich dann eine Zusammenfassung, vermutlich von beiden Vorträgen. Eine Kurzzusammenfassung von dem, was wir heute gehört haben. Und ebenfalls dann einen Anschluss mit den Überlegungen, wie kann man jetzt anfangen, mit diesen Kenntnissen für Varroa-resistente Bienen in den eigenen Völkern zu sorgen. Was danach kommt, werde ich euch dann, sobald ich es weiß, mitteilen. Damit sage ich herzlichen Dank fürs Zuhören für heute Abend. Wir sehen uns nächste Woche. Thank you very much, Stephen. We will close the session now. Have a nice evening. Have a nice day. I hope
Thank you very much.
I see most of you next Monday.
Okay.
Bye, Stephen.
Bye.